they can form a peptide bond. It's listed over here. And the peptide bond, yeah, basically when it's formed, you remove one oxygen from the left amino acid with a hydrogen atom attached to that, and also you take a hydrogen from the other side, and together, of course, you get water, because one oxygen and two hydrogen makes water, and you get a peptide bond, and it's actually quite a strong bond. So the cell can make this using certain enzymes, and they can make long chains out of amino acids. So they can, for example, start with the adenine, which is a very simple amino acid. It only has a CH3 side chain, and then it can link that to different amino acids. So it can make a long chain of this. And then the order of these amino acids is coded in the DNA. So DNA sets the order of these. And then when you have the long chain of amino acids, which is depicted here on the top, let's say you have like 10 different types of amino acids in a long chain. Those chains, they always tend to fold on top of itself. So they're first, of course, these molecules that don't like to be around water, they try to get close to each other, and the amino acids that like to be around water, they try to be on the outside of the molecule, and through those type of interactions, a protein always folds into a specific shape, and the shape determines its function. So in the end, it's the three-dimensional shape of the molecule that actually does something in the cell, and not just the sequence of amino acids. Of course, that's important because for a large part it determines how the protein folds itself, but the three-dimensional shape actually does something. And this process of folding, protein folding, that's actually a very complex process and not entirely understood, because it's, there are so many factors involved in that, so many interactions between each part, it's very hard to predict how amino acids fold. But once you have a certain shape, that shape often assists a certain chemical reaction to occur. So, for example, in this picture you see how two substrates, it's called, so the two compounds are linked together by an enzyme. So the enzyme would be a folded string of amino acids, and it brings together two substrates and makes a product. So it can also be the other way around, for example. So in the beginning, we spoke about how glucose is broken